Dr. Gustavo Baptista is a research assistant professor at Virginia Tech, specializing in molecular parasitology. Gustavo, thank you so much for taking the time today.
Yeah, thank you, Joe, for having me here.
So, to start, uh, could you tell us, the audience, about your research at a high level?
Yeah, absolutely. So I'm a molecular parasitologist, and my research focusing mostly tropical neglected parasitic disease. Uh, More specifically, I work with a parasite, Toxoplasma gondii, and there are few areas of specificity in my research. So I work with molecular parasitology as well as host parasite interaction. So how parasites interact with our cells when they infect us, and how they do gene regulations that are stage specific for each time of their life cycle, as well as some metabolic pathways. And then I try to use some technology-driven approach to answer some scientific questions like what are the essential pathways in the parasite that can be used as a future therapeutic target. So basically, that's in a broad general. I want to understand how cells of those parasites works um, in order to tackle and tell other research this is important and this is not important.
So, you said that your research focuses on molecular interactions between parasites and their hosts. What novel mechanisms have you uncovered that could lead to new treatment strategies?
One of the reasons why I'm interested in parasites is that when you go to research, biomedical research or biotechnology research, we do have studies in humans or other mammals like mice. So we know a lot about those type of cells or those species, but we know very little about parasites. And when we study those parasites, we see they do many things differently. And it's in that difference that we can find potential target. Because if the parasite using something that we don't have, a drug would probably be more effective in the parasite and have less side effect in us. One thing my discovery show is that if you get ubiquitination of protein, which is basically how the cells degrade the proteins, it's well known in humans, but it's much less known in parasite. And what I found is that the complex, those protein complex have slightly different composition than our protein complex, and therefore are potential therapeutic targets. For instance, my protein of my interest, we call FBOXL2, it's repressing genes to be expressed in the parasite, keep those genes off. But it does it in a different way, not targeting the transcription factors that bind to those genes directly, like they do with us. It's like an indirect effect. And in find this, I found another regulatory complex, and this regulatory complex is also present in humans, but there are some missing proteins in this complex, suggesting it can work differently. So now we can use drugs, some of those drugs, even the FDA approved drugs that are like disulfiram, it's approved to use in humans and can have a better effect in the parasites.
So, just backing up for a second, how do you define molecular parasitology?
Yeah, molecular parasitology is it's a field of study and research that focuses in parasite, but uses molecular techniques. We study the parasites at the molecular level because cells are living organisms, but they are made of molecules that are non-living. So we tackle like a smaller level of stud to actually know how the cells work inside, how the proteins, the DNA, RNA, they function and how they interact with each other.
So what is Toxoplasma gondii?
Yeah, so Toxoplasma is a unicellular parasite, so different than other worms. They has only one cell, and it's a apicoplexan parasite. It's inside a group of parasites called apicoplexans, and infects a wide range of animals. You can find this parasite infecting humans, other mammals like horses, even chickens, and you can find even in marine animals. So it's very distributed. So it has medical and veterinary importance. And besides this, we can find infections with this parasite in all continents of the world, different from malaria that we usually see in the tropical areas. Right? Fortunately, most of the infections are asymptomatic. People will get infected, but with good immune health, they can control infection and live healthy life, even if the parasites are inside their bodies. But in cases where our immune system get depressed, like organ transplantation, chemotherapy, HIV, the parasite can come back and grow fast and causing severe symptoms in our health, like encephalitis, an infection in the brain.
So you mentioned apicomplexan parasites. What are is it apicomplexan parasites? Is that pronounced right?
Yeah, apicoplexans are a group of intracellular parasites. So all species in this group must live inside the cell. They must infect the cell in order to stay alive. Some parasites can stay outside cells, but the apicoplexans are intracellular. And they all have some organelles in common, regardless of each parasite. In this group, you're gonna have Toxoplasma gondii, then we have Plasmodium, which causes malaria, and Cryptosporidium that can cause diarrhea. Right? Those are the three main examples, but we have many other ones. And those organelles are in common. Which organelles? One is called epicompoplast, which is basically like a chloroplast, like in the plants, but without the chlorophyll pill. And it's important to provide nutrients and plays a role in invasion and survival of the parasite in the host, like us. So that group has this complex of organelles at the apical end of them. That's why they call apicomplexans. They have a complex of organelles in the top of the cell. They have a topology.
You also mentioned earlier the stage-specific gene regulation. What is stage-specific gene regulation?
Yeah, so cells, as a living thing, they need to renew itself to stay alive. And the cell does it mainly by two ways: synthesize new proteins and degrade old ones. And in this process, the cell needs to know which genes need to be activated and which genes need to be deactivated. Depends on which part of their life cycle it is, right? So that's gene-specific regulation. What genes needs to be on now and what genes needs to be off now, and when I switch those. Right? And in Toxoplasma specifically, it can infect the cat where it does sexual reproduction. Right? And can infect all the other animals I mentioned before, including humans, where it does asexual reproduction. That balance when the parasite needs to know, should I go and undergo sexual reproduction or asexual, depend on which genes the parasite activates. And the proteinase stud that I mentioned before, FBOXL2, plays a role in keeping the genes off and making sure the parasite reproduces asexually when it infects a human, for instance. right? So if you take this protein away from the parasite, if you knock out the gene, right, the FOXL2, the parasite now start to activate genes that should be repressed, and the parasite now cannot reproduce properly.
You also mentioned metabolic pathways earlier. What are metabolic pathways?
Yeah, so metabolic pathways are biochemical process. They use those molecular machines like proteins, the DNA, RNAs, and how they work inside the cell, and allow the parasites to generate energy or to synthesize other essential molecules like amino acids or fat, right? And usually it's performed by a series of enzymes that controls the path of making, for instance, a molecule of glucose into ATP, which is the molecule the cells use to produce energy. So it's highly adapted, so the cells can activate different pathways and deactivate others. And that links to what our previous question was, like what genes are activated to make the pathway to happen, and what genes need to be off so other pathways don't interfere with the process.
So, how do parasites hijack host cellular pathways, and what implications does that have for disease progression and treatment?
Mm-hmm. So Toxoplasma is a great example of that, because once the parasite infects a cell, it evolved to detect that the cells will try to kill, right? And the way the parasite evades this response is by injecting some proteins inside the whole cell. And those proteins deactivate or activate other metabolic pathways or other genes to prevent the cells from killing the parasite. So there's examples like ROP proteins. One of those proteins, they go to the nucleus of our cell and prevent this cell to synthesize the effectors that will kill the parasite. And another way is that Toxoplasma are ingested basically by water, food, or contact with cat feces that are contaminated, right? So usually it's an oral or a foodborne disease. And the parasite start infecting the cells in our gut, in our intestines, right? But it wants to spread the cell throughout the body. But this parasite doesn't have flagella or cilia. It's not a very mobile parasite. So what it does, it infects a immune system called dendritic cells or other monocytes. And those cells circulate throughout the body, carrying the parasite to other places like our brain, heart, and muscles.
So, what challenges do you face when translating molecular parasitology findings into practical applications for veterinary medicine?
What challenges that there are many challenges. Sometimes we have technical challenges. We don't have a perfect model. In science, what is a model? It's a simplified version of a complex disease. Right? And sometimes we don't have a perfect model for that. So, for instance, in Toxoplasma in cats is a very complicated system to work, because in theory, you should infect the cats. And there's ethical implications of that. And we don't have a good in vitro system where you can use only cells to study the system. And even when you go to the in vitro system, where you study the cells in a petri dish, you don't have the full complexity of an organ, right? And then you have those technical challenges, and also many things we just don't know how they work. So it's like looking to a black box without opening, And only when you can crack that black box open, you can actually know what's inside. So we work in the edge of what is known and unknown. Sometimes we don't have the correct tool. The field has progressed a lot recently. In the past, if I want to study the function of a specific gene, the process was very long and unspecific. Sometimes you need to do a selection and isolate individual clones and take like weeks, sometimes months. But now we have other molecular techniques, much more specific, like CRISPR-Cas9, that has been revolutionized many fields, including the molecular parasitology. So sometimes it's just we don't have the correct tools or the correct models to study those less common organisms. But most of the research is done in humans. When we go to other animals, we don't have good models to study, good systems to use, and good tools that work in that animal or that species.
So you mentioned the kind of the tools, the technology. How is technology and specifically AI helping to advance this research?
Mm-hmm. Yeah, AI has been, it's still in the beginning, but it's been contributing a lot, especially in optimizing very time-consuming process. For instance, it can help with precision medicine, vaccine development, and diagnose. For example, diagnose, we can have a database of images, high resolution, high quality images, and train AI to detect a disease, a specific disease. So toxoplasma, in my case, right, they form cysts inside our brain. So we can get images of MRI or ultrasound or X-rays and use those images to say, this is a toxoplasma cyst. And we trained at the AI. And now when you have a case study, when someone comes here or a patient, even if it's not human, I'm in the veterinary school, so I can have some dogs or even cats. And I do their MRI or their X-ray and show to this AI. And now we'll be able to scan much faster than a human eye that cyst and tell possible Toxoplasma gondii cyst, right? Or possible tissue damage because the parasite is growing inside that brain, right? And then we can have a pathologist, someone that is specializing in looking at the tissues to confirm that diagnosis, right? We still need someone with expertise to actually confirm and not and prevent like false positive. The AI thinks it is, but it's actually not. But the point is the pathologist doesn't need to spend hours scanning those images now because the AI can do it much quickly and then it just check and confirm if everything was right. So it's just speed up the process of diagnosis, right? And also in the future, maybe developing new vaccines without that.
So you mentioned the future and how maybe it would help with developing vaccines. What is the future of your research and thinking on maybe a 3-year or 10-year time horizon? What does the future of your work look like?
Yeah, so that's a very good questions. Like for my research specifically, I found a protein, FOXL2, right? And it seems to be important because if I knock out this gene, right, if I remove this gene from the parasite now start to activate genes that should be repressed. So I basically remove a repressor. And now all those genes are on and they start conflicting with other genes and the parasite cannot replicate properly. So my research had described the process. This is how it works inside the cell. The next step, can we make this process a drug target? Now it's the next step. Like this protein is regulated by another complex, right? And there's a drug, disulfiram, which is an FDA approved drug for other disease that I can use and try to see if this drug can block the function of this complex. And basically having a possible therapeutic target. Since it's a FDA approved, there are many like criteria like safety and toxicity that is already established for humans. And now I'm translating that to the parasite treatment. And now let's see if it's inhibited or not. I still don't know because we haven't done those experiments, but that's what is the basic research it is about, is to describe something we didn't know, tell it's important or not, and now let's test if it can be turned into a drug target. So that's the next step that I will pursue. And also I work with this protein in a human cells. But since I'm in a veterinary school, I also interested in find the treatment for animals as well. And now, is this protein behave the same way in other animals, like in horses or dogs, right? So that's the other second step of my research.
Awesome. Well, Gustavo, thank you so much for taking the time to share your research and your insights, and I hope we can continue the conversation down the road.
Yeah, absolutely. Thank you for having me again.